Okay, thank you. So I think I just use about 10 minutes or up to 50 minutes quickly guiding you about if you want to apply our data. So this is about how to apply this multi-omic data. And just to talk about understanding society. And yeah, that's just overview what data available to you and where to get the data. And if you have question around that, I can quickly answer that. So I mean, Mina and Anna quite often use genomic, and sometimes if we also call it genetic, but basically what we mention here is just the SNP data. And for understanding society, the sample size for SNP data is 9920. So, just a little bit below 10,000. And we have this genotyped, which means physically measured, about 50,000 SNPs. And we got some 40 million SNP imputed. And the SNP data measured by this Illumina human core exome chip, and it performed at Sanger Institute. And it imputed using combined UK10K and Southern Genome Reference Panel. And the population I want to mention is white European, because sometimes we got some inquiry ask if you have minority group of genetic data, and unfortunately our data based on white European sample. Okay, the second omic data we have is DNA methylation. As Anna mentioned, we often refer it as epigenetic data, and we have two batches. The first one is measured in 2017. At that time, we got some funding to measure a relatively small subset. So we have 1174 individuals, about 857,000 sites after we had some quality control. For second one is we did it later at 2020, and for this one we have a relatively larger sample size, so 2480, and again we have similar GPD site after QC, and this 2-1, although the number of the GPD sites is slightly different, but the majority are overlapped, so you don't need to worry too much the difference. And basically we use Illumina ephemeral methylation epic arrays performed by Professor John Mill in Exeter, and the data normalized and cleaned in biology science department in University of Essex. And again, the population is white European. And as Anna also mentioned, we have 5 epigenetic clocks based on this methylation. So some subset of the methylation, and this related to age. And the third one is Protonic Data. So we have a 2-panel cardio metabolic panel. So we have the 6,180 individuals, 92 protein, similar for neurology panel, the same number of people and the same number of proteins. And it made by some company called Olink, and they use this proximity extension assay method. And I did some quality check for that, and we created some kinds of user guide. And for this data, we have 92% of white people, and we do have some other minority group have this data. So that's that. So that's basically 3 omic data we have, and there's 2 ways to use the data, so it depends on what you want to do. The first one is that if you just want to have genetic data or epigenetic data as your control data for some study, so they basically just want those information, then you can directly go to the European Genome Phonome Archive to download the data. The only thing I need to mention is that because it's deposited by different partners, so the genetic data and epigenetic data do not link to each other, and we were going to redeposit those data later, and in that case you should be able to link these two datasets. Of course, the epigenetic data is a subset, the sample is a subset of the samples of the genetic data. And also, you cannot use the link, the ID to link to any of the main survey data. On the other hand, if you just want to use proteomic data or epigenetic clocks, then we're going to make it available publicly. So, which means we're going to deposit in the data archive with our nurse visit data. So we're going to update that very soon. The second type of way of using the data is that if you want to link those genetic or epigenetic data to any of the survey data, then you need to apply for that. Of course, you can also link those data with protein data or epigenetic. So I give here the link of the our website to do that. So basically, if you click this link, you will find that you need to fill two things. The first one is your application form. So you briefly introduce what you want to do and give some summary and why you think it's important to your study, and list the principal applicants and anybody else that are going to use the data. And also in the form, you are going to find something like, do you want to use genetic data, epigenetic data, or do you want to have the imputation, and so on. So that's the information. Additional to that, you need to fill in a list of variables you want to use, which means the survey data. So basically, you will see it's somehow like a form, like the one I list here. So you should give something like what's the data file the variable comes from, and your variable name, and which wave it comes from. So from B1 to B18, it's just the BHPS data, and from 1 to 10 is our understanding society wave 1 until wave 10. Sometimes some people just give very raw description. So for example, I want any variable related to smoking, and that would cause trouble because we don't know what exactly you want. So do you want just to know, who's a current smoker, previous smoker or non-smoker, or do you want to know more about their history, when they stop smoking, when they start to smoke, or other thing. So in that case, if you give information like that, it's very likely we're going to contact you again, ask you to clarify which variable you need. So to save the time of your process, it's best you first go to the Understanding Society website to look at those available variable and find out the name and list of them. Of course, if you want to know, for example, what information available and so on, you can contact us before you start to make application. Okay, so briefly give you some idea about this data, or size of the data you're going to receive if you apply the data and get it approved and finally, receive data. So we have basically, we're going to use a way called Zen2, which is just an email sent to you with some link is a password protected way to send your data, but that's only send some data under 2 GB. And for the large data, we are going to send by FTP website with password protected. And if you just send this measured genetic data, then it's just up to 3 GB. So, once we zip it, we probably can send you by Zen2. But if you require any imputation data, then it will, up to 95 GB. So could be quite large, and it can be opened in R or Plink or some other software. And for epigenetic data, we have 2 GDS file, and it's up to 23.4 GB, and it can be opened in R. And for survey data, normally it's in stata or text format. We're more likely send you by Zen2. And if you also additional apply for proteomic data or epigenetic clock, those were sent by stata or text format. And basically, all of those data will be linked by a unique ID, but you will not be able to link to other survey data except the one you applied, and you should not try to do that. And I think I already mentioned, but I want to emphasize that if you just want to proteomic data or epigenetic clock alone, not with any genetic data or epigenetic data, you just go to the data archive to download it. So you don't need to go through this application process. And last thing I want to mention, we have some user guides for biomarker, which is online. And we're going to put the proteomic user guide online very soon. And also we'll have the epigenetic clock user guide online very soon. But for genetic and epigenetic data, we will send you some README file with more information about quality control, how we imputed data, and so on, this type of information, but we will not put it online because this data are available only through application. So we try not confuse people by just put some README file, but without the data. Okay, so last bit would be if you have some general question, you can contact Mina with her email address I list here. But if you really start to make application and want to know the progress and so on, or you have particular question about some variable and so on, you can contact the genetic team, which I gave the information here. So that's from me.